I'm interested in learning and memories. I'm interested in how our brain learns information today to be able to use it tomorrow to guide action. I think it's a very important question because memories define who we are. I am what I am today, most likely as a result of my actions in the past and what I've learned in the past. And what I learn today will most likely guide my actions in the future. First thing that comes to mind when I think about memory and information storage is a computer. In a computer, there is a specialized hardware storing information, a RAM or a hard drive, and another hardware separated from the previous one that actually uses the information and processes the information to have the computer make the actions that the computer needs to do. Differently than a computer, in the brain, the same hardware stores information and computes that information. The neurons, which are the elemental particles or the elemental cells that do the computations, also store information. And they do that in several ways. I'm interested mostly in how they change their structure according to the information they've processed, and how changing their structure helps them or changes them to allow new information to be processed in a different way. So specifically, I'm interested in synapses. Synapses are connections between neurons, and this is where information flows. And I'm interested in the basic principles that make synapses change according to the information that has flown through them, and how such changes change the way the neurons compute information in the future. Although information is stored in the whole brain, there are specific brain areas which are more involved in dealing with certain kind of information than other brain areas. I'm very interested in the hippocampus. And that's because the hippocampus has been involved in processing and recalling episodes of daily life. To understand the basic principles that govern synaptic plasticity, that is, the changes in synapses that allow information storage, it would be wonderful to be able to observe these synapses with time, and potentially in a model, an animal model, and we use the mice for that, in which one can teach the animal something, and the animal can learn that something, and the animal can demonstrate that it has learned something. Now, today, thanks to advances in light microscopy, it's actually possible to observe these synapses in living animals through time. However, the challenge for my research is that the hippocampus lays deep inside the brain. To give you a figure, it lays approximately 1 millimeter below the surface of the mouse brain. And so this has prevented anybody to look that deep in the brain until approximately 7 years ago. Now, during my postdoctoral stay in Mark Schnitzer's lab at Stanford University in the United States, I've developed a technology that allows me to place a tiny piece of optical fiber down into the brain without disrupting as much as possible the function of the brain, and to actually observe tens, thousands even, of synapses over the course of weeks to months as mice learn and recall memories. I tracked hundreds and thousands of synapses over weeks, and I was able then to compare the dynamics of these synapses, that is, how these synapses change over time, with the dynamics of synapses in other brain areas, such as the neocortex. This is important because the hippocampus and the neocortex are involved in different kinds of memories. Namely, the neocortex is thought to be storing memories for long amount of time, even for the whole life of the animal, while the hippocampus is thought to be important for very fast online information storage and short time recall. So for the first time, we could track synapses in the hippocampus, which allowed us to quantify how stable these synapses are with time. This is important because we think that synapses, or the stability of synapses, reflect the stability or the permanence of a certain amount of information in a brain region. In fact, the hippocampus has been proposed to only store information for a limited amount of time, which correlates perfectly with the stability of the synapses that we can measure. In fact, we find that over the course of approximately a month, 100% of the synapses of basal dendrites of hippocampal neurons completely turn over. High turnover means that if today I were able to look at the 1,000 synapses in the mouse hippocampus, in a month from now, I wouldn't be able to find any of them. Instead, a brand new, or at least partially overlapping set of synapses will be there, so that none or almost none of the original synapses will be there, and a brand new set of synapses will be there. This shows or can help understanding how information can be erased and it's only stored in the hippocampus for a short amount of time. The stability of hippocampal synapses correlates very well with the amount of time information has been demonstrated to be stored in the hippocampus. Additionally, we were able to compare our dataset with datasets acquired from other labs in neocortical areas. This has been very important because the neocortex is supposed to store information for long term. And indeed, in the neocortex, it has been shown that synapses are much more stable. This makes perfectly sense with our data, where the hippocampus has very fast synaptic dynamics that allow it to store information only for a short term, let's say a month in mice, while the neocortex has much slower synaptic dynamics, so there are more synapses that survive, they stay around for longer term, which allows this brain area to store information for a longer time, in principle, for the whole life of the animal. I believe my findings are relevant because they provide a cell biological basis that can explain why certain brain areas store information for a longer amount of time than other brain areas. Understanding how memories are stored given synaptic dynamics can allow us in the future to design treatments that target specifically synaptic dynamics in specific brain areas, and hence can help patients with memory loss, such as Alzheimer's patients, to keep their memories longer or even potentially to recover all their lost memories. This is only the tip of the iceberg. So far, we've been looking only at excitatory synapses, that is, at synapses that transfer positive information from one neuron to the other. However, there are also inhibitory synapses, which regulate very tightly the flow of information between neurons. Recently, we were able to label inhibitory synapses, and now we are tracking these synapses to understand the role of inhibition in structural plasticity of hippocampus. On the other side, the way neurons are connected through synapses changes the way they are active, changes the patterns of activity, in this case in the hippocampus. We have a method that now allows us to look at activity patterns of thousands of hippocampal neurons in live mice, and we plan on tracking those as well in mice learning and recalling memories, and to correlate activity patterns of thousands of neurons to connectivity patterns of these thousands of neurons in live animals.